either have some coffee now. We might just not have an after lunch session and just get started now and shoot the breeze about ideas. I think, but go, Oksana, you look like you want to have some coffee. Go ahead, go for it. But the idea is that we've got 3 microphones we can pass around for big, crazy ideas that we might be able to do as a community. And Eric and Andrea, you weren't here, but one idea is we want to do functional pangenomics instead of functional genomics. So forget functional genomics. It's so passé. It's so 2010. It's time for functional pangenomics. And I think we're doing really well in terms of mouse, rat, and human, probably Drosophila, definitely yeast. So I just want to solicit ideas for how one would do functional pangenomics.
Oh, I was just going to say, and zebrafish. Well, I don't know if I can add any more, but I think, so with all these genetic mapping studies, at least from what my lab has been doing, we certainly have a lot of locations mapped. And the next step, of course, is to identify the causal gene and to validate the causal gene. So in that regard, we are thinking of using more higher throughput approaches, such as zebrafish, where you can actually get access to the embryo and you can inject something and then get the gene knocked out. So zebrafish actually.
I was just asking, what's the generation time of zebrafish? Seems faster than rat and mice, right? Sure. No? Oh, for mice. Okay.
About functional pangenomics. I mean, I don't think it's very different than functional genomics, to be honest. Pangenomics just means you have lots of genomes. So now you have lots of references for the functional data you might acquire, which is pretty boring, but I think it may be that simple, actually. There's tools that can use the pangenome models for RNA sequence analysis. That's very interesting. And we've also done integration of ChIP-seq, there's ChIP-seq scanning systems that are based on alignments to pangenome. There's work that we've done on looking for motifs against the variation in population. I think all that is kind of interesting. There's, of course, epigenetic information that's layered across any of the sequences that are available that could be layered into the same model. And yeah, I think that there's things to be done, but it's actually not, from my perspective, it's not super impossible. You just need to use the different references from the genomes in the pangenome that you're working on, and integrate them together using the alignment of those genomes. So.
Yeah, I'd like to follow up on that as in terms of the functional elements of the genome. Especially for rat, there's a lack of data in terms of these functional elements. For rats for mice and human, there's ENCODE, and for rat, there's almost no data out there. So any data towards that would be great.
What I can add. It's true that pangenomics probably is still not fully ready to go downstream. We still miss the link between your sequences and the metadata. That's true. And I think that's one of the main focus that we'll have in the next upcoming years. But I truly think that it will come. Yes, too.
Yeah, I would have said the same thing, but also, yeah, it's actually so the connection between I mean, there's no tools at the moment actually to connect the phenotypes to the pangenome. So that's what I see the major obstacle. And plus, there's not enough data to build genotypes. So that's an invitation for all the people here who has not yet contributed to contribute to long-read sequencing and data accumulation. I'm probably talking to the wrong people because you all contribute, but that's the idea. So I just think there's not enough sequencing data to build pangenomes in first instance in model species, and second, the tools for connecting this data to phenotypes. So that's something that's interesting. It's you can think about in 1 year about having this as a topic in your conference. Could be like a mini session where but rather than presenting, it has to be like a workshop, like something, what's going on and how could you combine these for making efforts for funding this research.
There are a couple other topics, but I just want to come back to yesterday's talk by Anna about the cube idea. Basically, it's way more than a cube, it's a polytope. So, Matt, can you represent that? I hope some of you saw Anna's talk yesterday, but it represents a big effort by many individuals at JAX. I guess it's primarily out of one lab, or is
so the cube initiative was a multi-PI effort, but it was actually they set up kind of a project manager that ran data for all of the, I think, 12 projects. So I think it's fair to say it's one of the first attempts to do kind of institution scale science at JAX, as opposed to, 2-3 PI center grants. Now this was sort of a 12 PI effort. The data are going to become available publicly very soon. And it wasn't just on the diversity outbred arms that Anna presented. There was also a CC-RX cross with, lots of replicates, diet interventions, and so on. And then there were, expensive assays in 3 of the DO founders. And so the idea was to do kind of vertical and horizontal integration across genetic diversity versus deep phenotyping. I think it's fair to say that we're at a stage where we're finishing up some papers and trying to get, the keystone publications out and then deliver the data to the community to actually do good stuff with it. So, I wonder if I could say more about the organization of the efforts. So we started with the type 2 diabetes concept partly because Alan Addy's team had 500 DO mice from which they biobanked several tissues, several organs. So liver, so they had done the islet gene expression where they meticulously dissected out islets from pancreas. If that's your idea of a good time, God bless you. But they saved livers, hearts, adipose tissue, and what's the last one? Skeletal muscle, gastrocnemius muscle from the mice. And so we had access to all of those tissues, and we used that as kind of the ready-made rather than growing up a bunch of DOs and performing the challenge. This was supposed to be a pilot for other institution scale science. I'm not at liberty to say what those efforts are. I'm being slightly glib, but you could probably guess. But we tried to use it as a way to learn what kinds of things do and don't work at the level of institution scale science, and I'm nearly certain that it's public now that JAX just got a very significant philanthropic gift to build a physical presence in Portland, Maine for a data science initiative at the lab. I realize this is being recorded and I might be speaking out of turn, or is it not being recorded? I don't know if it's being recorded. Anyway, I don't want to get yelled at if that's not true. If the announcement isn't public yet, I don't know what to do. They can fire me. But there is a significant there's significant ambition in terms of data science and this kind of mouse human, vertical horizontal integration. Robin spoke to that a little bit as well. So many of you know Alyssa Chesler. She has a new role, or she has an expanded role at the lab as I won't be able to remember her role, but she's advising on this data science initiative. And Jack's just hired a new chief data science officer, an inaugural chief data science officer. So hopefully we'll see more efforts of this type in the future. But yeah, that's the cube in a nutshell.
David and I, and actually quite a few people, have been dreaming up a very large dialogue cross, and I think David's thought the most deeply about this. So it's basically a data cube also, or we're stumbling towards implementing this, but why don't you just.
Yeah, the idea is that we've got, say, 120 BXDs that are easily available, and the F1s make 120 by 120 matrix, all of which are isogenic and replicable. So now we already have all of those sequenced. So we have the genomes, we have many phenotypes on sort of the diagonal of the 120 inbred BXDs, and we can fill in anywhere in that F1 that we want. So you can phenotype 100 isogenic strains and then make predictions and test that in a different set of 100 that are related, but you've never seen before. So it's really a sort of mechanism for the idea of sort of precision medicine and predictive medicine, is that you can make these predictions in one population, test it in another, and we can have that kind of cube idea that now you have any of these many strains in any environment across any lab in the world, and we can compare these things. So it's really an extension of what Rob's been trying to do with Gene Network, of bringing all this data together, but trying to get around that limitation of there's only a finite number of inbred strains, but when you start breeding them to each other, you just get a huge matrix of hundreds of thousands of replicable animals. So the small thing we're doing at the moment is longevity in some of those, because we have longevity in many of the BXDs. So we're now testing those F1s to look for sort of hybrid vigor type phenotypes, and whether additive and dominance effects. So whether when you cross a long-lived by a long-lived, do you get doubly long-lived? If you cross a short-lived by a short-lived, do you get better, worse? What happens, basically?
And the cool thing is that with the rat resources, we can basically do the same thing. So with 100 inbred strains of rats, we're up to 200, 300. I forget how many we've sequenced at this point, but it means that kind of dialogue cross can be exploited. We had one talk by Richard Mott about all of the dominance that we're leaving on the table when we just use inbred strains. So it'd be great to rescue that. It's a tremendous signal. But if you do the dialogue cross of inbred strains, you have a lot of the advantages of Theo and HS with the replicability, which means, your great-grandchildren could potentially be studying the same genome plus, maybe 1,000 de novo mutations, which, as I mentioned earlier, those de novo mutations will be hugely helpful because it'll be trivial to get T to T on anything from a tail snip in 10 years or maybe even in 3 years. So, I think that the idea of just doing things by forward genetics or just doing reverse genetics using the knockout, that was frankly passé 10 years ago. But I think our community as a whole has not grabbed onto the idea of doing reverse genetics with classic forward genetics resources. But we really have to change our mindset and just think of both simultaneously. And that's where the mediation comes, because you can mediate from both directions. You can say, I have a mutation in catechol methyltransferase. What should happen? And there you have a ton of biology. And then you can ask, well, what traits map to catechol methyltransferase? And that kind of analysis becomes incredibly valuable. So we can do it for rat, we can do it for human maybe soon. And we can certainly do it for mouse. And it's great to have these efforts where people realize it's really, it's industrial scale, institutional scale. It's more like a Manhattan Project. If you wanted to do it properly, you'd say, give us $1 billion, please. Maybe Elon Musk or somebody, some maniac, and we'll do this right. And we'll not only have development, we'll have aging, we'll have the colony rack systems, we'll have the systems that Vivek showed us yesterday. We'll have just a huge panel of tissue that sits in a biorepository. So if you want an embryonic day 13 of this particular genome type, you can get it. If you want a 900-day-old animal treated with rapamycin, you can have that too. And it would just be an amazing, basically, if you want to reduce animal resources, if you had this flurry of animal production, and I think it just could be transformative. And which brings me, Shelby, to put you on the spot wherever Shelby is. Did he escape already?
I think he's escaped.
He escaped?
Yeah.
Okay. Well, who wants to represent AI? Because this is going to be an AI problem. I mean, the work Matt did, showed us with mediation, fabulous, but it requires too much painful thinking. And the way is just to throw all of this at a deep neural network and figure out how to get the AI system to provide some insight. Who wants to take it? Eric, you're the only person I see here that's doing a lot of AI work now. So, and how, if you don't want to field it.
We're talking about integrating lots of data, especially with functional information from many experiments, which we haven't been able to do yet in these models. Okay. I guess there's just many ways, like these systems, these are machine learning systems. So you can use them to compress data sets and build generic models of what's going on that might integrate DNA and functional annotations. There's only been a little bit of work on this done at all in any context, to my knowledge. For folks who are building systems that represent DNA sequences abstractly and then using them for classification tasks, like this is an enhancer, this is a gene body, this is a long non-coding RNA. So you have this kind of stuff going on, and I guess it should just work if you have the data and the resources to do the training. The utility of it downstream is a little bit questionable. I think pretty much every project could, to some extent, end up with its own model, and so it's really just a collection of techniques that are a bit like statistics, but updated to fit much more complicated functions that you can then interrogate to the data sets you have.
Yeah, so I pulled down a bunch of data of relevant traits, behavioral traits, brain anatomical traits from Gene Network, and I did some PCs, and I tried to look for outliers on the periphery, sort of, smarter than average, dumber than average. The shameful secret that I didn't mention in my talk is that the reason I didn't get to do more BXD crosses is because I failed to recognize that the only commercially available TSC knockout mouse is a conditional on a mixed background. So I had to do a back cross, and that burned up most of my DoD money doing the back cross, and it burned up a lot of our time. And then the pandemic hit, so we really had that one shot at one BXD. So I had several others that I was going to test that I'd sort of selected from the periphery of this Gene Network point cloud. And then the dispositive feature was that we were running out of time to make the order, and BXD87 was going to be available on time. So of the 6, I said, bam, that's the one. And yeah, I'm curious to see if 50% of the BXDs, if when we make the F1s, we'll have seizures, in which case, yeah. Yeah, was I really lucky? Or was the construction that was going on right next door, somehow stressing out those mice and causing seizures? I don't know. But yeah, how did I pick BXD 87? It was a mix of very weak evidence. Yeah.
Can you predict that the CA transversion frequency should be greater in BXD 68 ab initio? It should be. We know it is now. It's a BXD that happens to have a couple of missense variants in muddy H or muddy Y, muddy HY, whatever. But that might be a way to kind of get it.
I mean, the question is if your N is big enough that you can actually learn models that somehow incorporate all these things so you can go forward. Do you guys think it is that, like, is there information available latent in the data that really could have inferred that, or is it something that requires external knowledge of the gene systems and function? Like, if you want to just learn, you have a bunch of phenotypes and a bunch of BXD crosses, right? So do you have enough to actually learn a model over that you then can go forward with? Yeah.
I think you probably need that external kind of data, that external knowledge. I think there's a huge amount of data on gene network, obviously, but there is a lot of not well-recorded metadata. So I don't think you could just throw everything in gene network at an AI and say, make predictions without sort of being able to parse out, okay, this was done in 4 months olds, that was done in 24 months old, that was done in males, that was only done in females, that was done with an N of 1, that was done with an N of 20. I don't think that's one problem. And I think really we need AI that knows some biology, because kind of if one of us says, we know there's relationships between different immune functions, I think any AI is going to need to know that to be able to make sensible inference, me knowing very little, if anything, about AI.
Yeah, I just want to comment here that we do have a lot of information, right? So there's a lot known about biochemical networks. There's all of this stuff is formalized in databases. The AI does know that, or should know that. Yeah, and I'm thinking about resources like Reactome and KEGG and so on. I mean, these are machine readable resources that tell us what protein interactions work at minuscule detail. You can go from high level interactions down to phosphorylation, ubiquitination events. All of this can and should be built into all of the models that we're looking at, right? This is not really black box anymore. So, I mean, an AI that's got access to that ground truth, it may still be incomplete, but it's still a lot of data there that is potentially informative and is certainly going to constrain a lot of the models, so that we're not fishing in the dark.
I think it was at CTC actually when Abe was hosting several years ago in La Jolla. Trey Eidecker was invited to give a talk, and he had just published a system called D-Celled. I don't know if people were in town when he showed that, but he used the big single and double knockout compendium in yeast, and he trained what's now in terms of artificial intelligence considered very lightweight deep neural network model. And the innovation that I thought was exciting and hasn't been followed up in multicellular systems to my knowledge is this yeast compendium has single and double knockouts and growth phenotypes, and they trained two separate neural networks. One was sort of their baseline option where they just did a standard architecture. Can I predict from a series of these layers, can I predict the growth? And the answer is yes. And you get some black box representation of the growth phenotype internally to this network. The other option was that they actually structured the neural network according to the gene ontology, where they each the input data was a binary representation of what genes were knocked out. So you had a bunch of genes on, and then you put zeros where they were knocked out, or vice versa. And each of those genes was annotated to a lowest gene ontology term, which is annotated to higher order terms, and on and on and on. And very near the top of the Go hierarchy for yeast is growth. And they trained that neural network. From the point of view of a neural network architecture, it was just a big, deep architecture with a funny shape. But what they showed, which I thought was actually kind of astonishing, is that if you looked at the quote unquote activity level of neurons high up in the gene ontology, that corresponds, of course, to a set of genes that we use to annotate our enrichments. And when they looked at the expression of the genes that are annotated to these higher order pathways and looked at principal components of the measured expression of those genes, it correlated with the activity level of the neurons at those levels of the hierarchy. So in the process of going from knockout genotype to this one phenotype, it somehow, that network had to at least learn some kind of surrogate of the molecular physiology of that trait. And I think it's, at least in principle, possible to use a strategy like that. We have phenotype ontologies. Most of our, it's not impossible to annotate, gene network. It'd be a Herculean effort, and I think some of it's maybe done. But you could try something like that with phenotype ontology, and maybe put ancestral haplotypes at the bottom instead of knockouts. But a strategy like that, I think, would be a potentially elegant and principled way to bring in some of these computable ontologies. I don't know how you would do it with KEGG. In principle, you could do something similar with KEGG. But yeah, I think that, I've tried to twist people's arms to do it and give me credit, and to no avail.
These ontologies are typically, they're graph structured, and so graph neural networks would be a natural application. Rather than building the neural network to match the topology of the ontology, you would just, you'd be modeling a graph in effect that had
fair. Yeah, fair enough. Yeah, how you would map the genotype into that graph, I think, would be the interesting piece. But yeah, in principle, yes, I totally agree.
Yeah, I think the single cell correlation networks that Laura Sabers group showed today will be a huge help in this kind of effort, where you have single cell types now, and if you have the genotype permutation mapped onto these networks, then it will be very helpful information.
Yeah, what might be a cool and easy thing would be just to be able to predict gene expression in different BXD strains. So we've got a huge amount of molecular data of sort of what genes are linked to each other. We know a huge amount of sort of eQTL data, and we've got a huge amount of different transcriptomics, proteomics data in gene networks. So say I've not looked at the expression in BXD87 in the brain, but can I predict what the expression of X and Y would be in that strain we've not looked at? So that could be a interesting thing. And I think the molecular level, like you say, is possibly easier than the sort of phenotype level, physiological level.
I don't know if this is, he's handing it off.
I have a completely unrelated point, so you should finish.
Oh, I was gonna change gears completely. But speaking of the diallele cross, and you said, and Rob, you mentioned that there's this sort of perennial push to move away from animal models, and in particular, the organoids are coming along, and they're getting pretty good. They're not organisms, but one thing that I haven't seen anybody attempt to work on, but is in principle doable, in the context of a diallele cross, is you could in principle make chimeric organoids. And if you care about developmental phenotypes, you could look, for example, and sort of mix and match progenitor cells from different founders and look at organoid competence, chimeric embryo competence, and actually map out factors in those early developmental stages, which is actually a major breakthrough compared to what you can do with the whole organism. And so it seems like finding ways for our community to contribute to that organoid world would be an easier way for them to, consider us valuable rather than, put ourselves in opposition to them.
Rob asked me to ask a question, so I'm going to do that. One, a lot of this is over my head, so please forgive me. But I think a lot of the problem is that I know at least 2 groups at Madison that have BXD data that we haven't put anywhere. And I guess, how do you encourage people that aren't part of this specific community to share and also help people? Because like, I have no experience really in QTL. I've done a little bit by myself, but I didn't really even know that much about Gene Network when I got here. And so there's probably lots of resources as well that aren't being shared. And I think it's in every field, it's a problem, but I don't know how you kind of fix that, or if anyone has any ideas.
Yeah, I don't have an answer, but yes, and it's not just that people aren't sharing the data, it's that tools get replicated again and again because people don't know that it's already been done by someone else already. So yes, and we talked a little bit the other day about some APIs and being able to share data and tools between different websites, which I think is great and is definitely seems the rat community are getting better at that and sharing data and sharing tools. I'm not sure how good we are at, we're trying to be better in the mouse community, but there's still sort of little silos of people want their tool and are very protective of their tool. And I don't have a good answer to this. I just agree wholeheartedly that we need to be able to get people together and get data in shareable formats. So like we say with the APIs, it doesn't matter where you put it as long as you can get it to everybody else and they can pull it into their favorite tool.
Yeah, I think in an ideal world, there would be, rather than trying to get people in the community to push data, you'd actually have some person to go out as like a rep and pull the data in. This is a big curation problem. Crowdsourcing curation is very hard because people are not curators, and I think it's kind of a curation issue. And I wonder whether it was possible for the community to potentially find a way of funding that. Yeah, I mean, but if a lot of people are saying this is important, could you actually get funding for like a meta work group that could just be essentially curation for the community?
I definitely think that'd be great if someone could fund it. Someone just basically looking through papers and going, hey, look, that information would be useful. Send them an email, just give us your data. Was the question over here, or I just see someone stretch?
No, now I'm over here. No, totally agree. We at the Rat Genome Database, we do our best, but we have a limited number of curators. There's only so much that we can do. But we're trying to come up with ways of helping the community so that, because the problem is that when you have 100 people in the community who are trying to do curation, they each do it their own way. So you've got 100 different ways of standardizing, and it never ends up standardized. So that's one of the things that we're working on. The Alliance of Genome Resources is also working on it quite hard to try and come up with ways that we can help the community to really be more standardized in the ways that you express, that we all express the metadata, because the data is key, but if you don't have the metadata, you can't find it and you can't reuse it. So the metadata is really another key component of trying to get this stuff out and make it reusable and make it that, you don't have this one-off thing and nobody ever sees it again, nobody can ever use it again, but you've got things that are reusable.
Yeah, 100% agree. I also think over time, I think if you've got 100 people to do it 2 years different from each other, you'd have 200 different standards. And yeah, we sort of make a big deal about sort of the inbreds and the isogenics, that the great advantage of them is you reuse that data, but if it just stays in someone's hard drive or has a whole bunch of metadata that nobody knows about, they're not reusable. So we're losing half the reason we actually use these isogenic strains.
Hi, this is Laura Saba online. Can you hear me?
We can hear you.
I was just wondering, maybe Rob, I'm trying to see who's in the room by the video, but Rob, can you talk a bit about how many people do you have that input data into Gene Network? Because I know you have some tools where people can input it and then run their analyses, and some of those data tools for self-input. What is there a big group that uses it, or is that something that hardly anybody uses?
So people do put their own data into Gene Network, but a lot of the time they email the data to Arthur and ask Arthur to put the data in, especially with sort of big data sets. But yes, most of sort of what I call phenotype data, so the real trait data, people put that in themselves. We have got guidelines of how we would like to do it, but I don't think we have any way of sort of forcing those guidelines. So often it's someone puts the data in and then Rob goes in afterwards and shouts at them or redoes it himself. So yeah.
Because I mean, at least, so that mechanism, you have this carrot of, right, that you would be able to use all of your genotype and mapping tools if people uploaded it. I'm just wondering at what are some other carrots to get people to share their data besides getting to use different tools?
Yeah, I think that is a problem, and Gene Network is still struggling to have a good solution. I think the group from Paris that showed us what they're doing at the Pasteur Institute for the collaborative cross animals that they have in their group, that's where we're heading. We're almost there. But I think those are two examples of authentication and authorization with levels. Just like if you have Dropbox, you own a folder, you can share it with different people. They have different privileges. They can overwrite, erase, or they can just read. And we should be there real soon. Now is the famous catchphrase, which means maybe in 10 years. No,
well,
really, it'll be real soon now.
Well, and I like RGD's idea too, the thought that was brought up about these new data management plans being so unwieldy to be able to have, that's a great carrot. Here's the text that you would put into a grant, and keeping track of that to recontact those people if funded or something like that.
Yeah, make something like that available to people, and then they have kind of committed that they will put the data in. Yeah.
You up there on the screen, do you want to just tell us what next year is going to be like if you're around, actually there? Ah, there it is. Thank you. Okay, problem solved. Okay, so that's for next year's meeting.
What were you asking me? Sorry, this is Ann.
Yeah, go ahead and tell us a little bit about how you think it'll be structured next year.
Can you see me? Hi. I'm looking for dates where what I'm thinking is that we would do it sort of maybe a Wednesday start in an evening with perhaps a keynote or a session on a Wednesday evening, sort of like the Cold Spring Harbor style. This is sort of what we had done earlier, and it got people in to be able to travel and get there, in the evening, and then go through Saturday noonish. I like talks, but I'd also like to be able to provide a poster session if we can. And I'd like to also, I'm thinking about if we could have a sort of a Shimayama memorial lecture in honor of Mary. We were hoping to do this actually back in 2020, just before everything fell apart. So, you know, we'll do the hybrid format. Looking at venues, we have a structure now at MCW where we have to go through permissions and a couple different processes to be able to have on-site events. But I don't see that to be a problem. It's always just more of a problem of getting a venue. Because the medical students usually, well, always take precedence. So I won't be able to book a room probably quite yet. And that's what I'm looking into, is how early can I actually get a venue set. But we have a couple in mind. So looking at September, October, this seems to be a fairly good time of year, hopefully. And it would be great if people have major other meeting conflicts that you let us know earlier than later. And I'm open to feedback on what you would like to see the structure be.
More short talks. That's the only feedback I'll have.
So you prefer everybody talks rather than?
I think it's particularly important for younger people to have the opportunity to talk, even if it's somewhat exploratory, or even if it's almost like, here's my proposal for my PhD. What do you think? We've had a couple of those, and I don't mind them at all.
You know, we've done things like data blasts where, you know, they get up and basically have their poster deconstructed and give a really short 5-minute, even 5-minute enticement for them to people to come and visit their posters as well.
Yeah, those sort of elevator pitch kind of sessions.
Yeah.
Talks, yeah. One thing I was going to say is we'll try and invite people not from our community. I think we've kind of got a little bit inbred that we have kind of the BXC people here and the rat genome people here, but there's nobody doing worms or flies or yeast or sort of that do a lot of the similar kind of techniques, especially genetically, that we do, and that we kind of just don't get in. So maybe we can try and all think of someone we know that we can persuade to come next year.
That's a great idea.
I also like set aside times for discussion too. So it's not just all packed with presentations in a couple minutes, but actually some panel discussions about the direction of the complex trait research and what's new and what's hot and what do we need to come together as a community to promote.
Yeah, I agree with that. Maybe we should have done this session earlier in the meeting, maybe even on a Tuesday would have been ideal, at least to get people thinking about it. I would like us to think about doing some kind of group review paper on functional pangenomics in the next 3 years. I think we'll definitely be there as a community. I don't think, to the best of my knowledge, we're in an ideal position to do this for rat and mouse, and maybe human. Although human, there will be a lot of people doing that, so that'll be more competitive. But for rat and mouse, we're pretty much it. The other thing that just one more big, hairy, audacious idea. The methods that we use to evaluate the impact of sequence differences are still pretty primitive. But with these new, crystallography methods and AI-driven predictions of protein tertiary and quaternary structure, it should be, in principle, easy if you have access to enough GPUs to simply work through all of the variants crashing around in the collaborative cross, the DO, the BXD, the HRDP, the BXH, HXB, the LEXF, and just say, here are all the crazy mutations that if you don't see something, either our model is totally wrong and AlphaFold, AlphaSNP is screwing up, or you're just not looking correctly. And, I think that's one, we actually put in a proposal for $20 million that got turned down promptly to Oak Ridge to do this, but I think it's definitely worth doing because with the sequence quality we have now for mouse and rat, it should be a no-brainer to do that. And then we can kind of address this problem of, can we predict phenome from at least one genotype, one mutant genotype? And that's why I'm so keen also on getting the structural variants, because those are going to be definitely worth looking at. So if you have a duplication or an inversion or a whopping big, sign or line that's sitting right in the middle of a promoter region or 3' UTR, should be pretty obvious that something should be messed up. Flavia, I'm speaking to you. Okay. All right. People to thank. I want to thank Arthur, who did a great job organizing everything here. And yeah, and Tamara, who just came in, thank God. So, and John, who just came in. And John's going to tell us we have to eat now, which is great. And I want to thank
Richard
Flavia.
outside, who's been signing you in. Thank you very much. Flavia, who organized a huge amount of things for us. Yeah, thank you all for hanging in here.
Thanks, everyone.
And thank you
Thank
all
you.
for hanging around.
Sorry I couldn't be there the last day. Safe travels.
Stop recording.